Good afternoon. My name is Teresa Imperato, the ALSA clinical specialist here at the Greater New York chapter. I hope that you're all enjoying this year's Educational Resource Summit. Before we start, I just want to remind you that there's a Q&A icon at the lower section of your screen. Click there and type any questions you may have. We will do our best to go through all of the questions at the end of the presentation. I wonder if any of you out there remember the Wheel of Fortune commercial. What would Vanna wear? That's sort of the chatter at research conferences when we knew that Dr. Bedlack would be attending and speaking. What will Bedlack wear, we used to always say. I see now that he's inspired teen idol Harry Styles. Congratulations on that. Dr. Bedlack is not just known for his extraordinary sense of style, but also for his compassion, kindness, and devotion to ending ALS. Born in Connecticut and educated up and down the eastern seaboard, he ended up at Duke, where he completed his medical internship, neurology residency, and neuromuscular fellowship, and his master's in clinical research science. He has won awards for teaching and patient care, including Best Neurology Teacher at Duke, Healthcare Hero, Strength, Hope, and Caring Award, America's Best Doctor, the American Academy of Neurology Patient Advocate of the Year, and the Rasmussen ALS Patient Advocate of the Year. He has received research grants, participated in ALS clinical trials, and published more than 100 articles on ALS. Dr. Bedlack is the leader of the International ALS Untangled program, which utilizes social networking to investigate alternative and off-label treatment options for patients with ALS. The leader of the Reversals program, or Reverse ALS program, which attempts to understand why some people with ALS recover from it and to make this happen more often. Today, he's here to educate us on these alternative therapies. Welcome, Dr. Bedlack.
Thank you so much, Teresa. I've never had an introduction quite like that. Wow, Styles, holy cow. Just gonna pull up my slides here. All right, so greetings from North Carolina, everybody. I'm gonna talk today on a topic which is near and dear to my heart, Untangling the ALS X-Files. And on this first slide, I've got some disclosures just so you know that I have some research support from the ALS Association, from some companies, Orion and Medicine Nova, and from the Healy Center. And I've got consulting support from several companies that are listed there. Mostly what I do for these companies is try to help them design more patient-centric trials and help them with what's called data safety monitoring boards, trying to make sure the trials are as safe as possible for everyone who's participating. So I hope that people on this Zoom meeting remember the X-Files. It's been a while now, but it was one of my favorite TV shows. It ran for most of the 1990s. It made a little comeback from 2016 to 2018, which unfortunately wasn't very good. And then there were a few movies. But the show revolved around a character by the name of Fox Mulder. So when he was a child, his baby sister Samantha disappeared, and he was supposed to be watching her. And the way he remembered it, she was kidnapped by aliens. But of course, nobody believed that. And so that skepticism just fueled his fight. He spent the rest of his life trying to prove to the world that aliens and other supernatural creatures were real. And every episode of the X-Files would open up with some strange occurrence, and Mulder and his team would investigate, and they would usually find some rational explanation. So I kind of had my own Samantha early in my career. This was a 60-year-old university professor. Unfortunately, she came to me with what appeared to be ALS. She had just a little bit of bulbar weakness. She had a little more arm and leg weakness, but was still independent in her activities of daily living and able to walk with a cane. And she had no obvious cognitive or behavior problems. And so I sat down, I had the difficult conversation with her about the fact that I thought this, it was what she had and what that meant. And then I presented her with stage appropriate, evidence-based treatment options, including riluzole, and multidisciplinary team care. And then finally, I asked her if she wanted to be in one of my open research studies. And much to my surprise, she opted for the first two. She said, sure, I'll take the Riluzole and I'll come to clinic, but I'm not going to be in your study. Instead, I'm going to try something I heard about on the internet, which was called Dean Kraft's energy healing. And this is a very smart woman. I asked her to explain to me, why this was the direction that she was going to take. And she showed me this video. I'm going to show it to you now.
In 1985, Nelda Buss was diagnosed with ALS, amyotrophic lateral sclerosis, better known as Lou Gehrig's disease.
It was my birthday. I was 43 years old, and they told me I had Lou Gehrig's disease. Well, they actually said amyotrophic lateral sclerosis, and I still wasn't real sure what that was.
ALS is a medical mystery. There is no known cause and no known cure.
The established medical profession, they just said, goodbye, see you, have a good life, live it to your fullest.
The disease inevitably progresses from partial to complete paralysis of all the muscles in the body.
She just kept getting progressively worse. And I guess from the time she was diagnosed until she was basically an invalid, it was only about 6 months or something.
In the final stages of ALS, Nelda had become a quadriplegic, unable to move, but mentally alert and aware that she was about to die.
I gave away most of my good clothing that I had, and I had my dress all picked out for what I was to be buried in.
Then Nelda heard about Dean Kraft.
And it was just such a relief to talk to him. I said that the doctors say there's no hope. And he said, that's not always true.
Kraft did his laying on of hands for 3 months, and then they had a small but significant breakthrough.
In May, I was showering me and I could move my toe, and that was so exciting.
After her toes, Nelda slowly regained movement in her fingers, then her back muscles. Soon she could control her hands, then her arms and her legs. Nelda worked very hard on her own physical exercise regimen, in addition to treatments from Dean Kraft. Her family videotaped her progress over the course of a year.
It's wonderful to see her and her family being together and enjoying life together. Words cannot express the feeling that one has when this happens.
Okay. How's that?
Everything has changed. She's back and she's doing more than she ever did before. I mean, it's great to have a mother. It really is.
So I hope you'll agree that that is a pretty amazing video. And for the purposes of this talk, Dean Kraft's energy healing is an example of something that I call alternative or off-label therapies. I will abbreviate those as AOTs and define them as treatments advertised to slow, stop, or reverse ALS without what most of us would consider to be very good scientific evidence. Now, Samantha's not the only one that's interested in AOTs. It turns out that most people with ALS will try at least one over the course of their illness. Why? Well, unfortunately, the treatments that we have, much better than what we used to have when I started my career. And even those that are coming out in the next few years, they look like things that will improve quality of life and slightly slow progression. We don't have that home run yet that we're all looking for. And so, like the slogan on this old X-Files movie poster, patients and families want to believe that there's something better out there. And if you ask them, 10% say they think they'll find a cure by trying these things, 20% think they'll find something to make them better, 50% think they'll find something that will slow their progression. And onto that difficult background comes the internet, which makes it easier than ever for people to find AOTs. If you just pull out your computer or your phone and you do a Google search of the words ALS treatment, you will get double-digit millions of hits. Now, not every one of those hits is an AOT, but many of them are, including some of the first ones that usually come up. The other thing about the internet is there's a tremendous breadth of information, but there's not a lot of depth. There aren't any internet police that go around looking at the claims that people make and hold them accountable for those claims. And there are many extraordinary claims made about Aots for ALS on the internet. Here's some examples. These are from a web page that I believe is no longer active for a product which was called MotoTab. And you can see here at one of the blue arrows, this was claimed to be the world's most relied upon and clinically proven natural treatment for motor neuron disease. And on the right-hand side, clinically proven, it's completely guaranteed to work. You're at no risk from trying this. So what kind of evidence is offered to support claims like these? Well, in the case of MotoTab, there was absolutely none. Had you clicked on any of those links, you would come to something that said, clinical studies will soon be posted, and they never were. Far more common, though, than that scenario where there's zero information is a scenario where anecdotes are used to justify these kinds of claims. So anecdotes are short stories about a person, in this case with ALS, who tried something, and in this case had something good happen to them. And they're by far the most common type of information used to support AOT efficacy claims. And so that video that I showed you on the Dean Kraft website, that's an example of an anecdote. Here's another one. This one comes off the website for a pharmaceutical company called Genervon. They are or were developing a product called GM604. And it says here, after reviewing this patient's medical history, it's in the treating doctor's opinion that they've greatly benefited from GM604 over the past 6 weeks. During our initial assessment, the patient had rapid tongue fasciculation, but this decreased dramatically with the medication. The patient stopped biting their tongue. This is a reversal, an unexpected benefit. Moreover, their limb weakness progression has completely plateaued. Okay, well, again, this sounds somewhat interesting, but let's look at it a little bit more closer. There's some potential flaws in anecdotes like this. First, we don't know who this person is or how their ALS diagnosis was confirmed. And there are at least one example on the internet of companies having just completely fabricated anecdotes to sell their treatments. This is a cartoon, I believe, from the New Yorker that kind of illustrates this. On the internet, nobody knows you're a dog. So this was the case of a company called Lifestyle Lift. They made facelift products, and they had these before and after photos that looked amazing. And when they were sued because somebody got the product and it didn't work, in court, employees of the company said they were just told to basically fabricate the after pictures, and the company wound up having to pay a small fine. Another problem with anecdotes is that they tell us about outcome measures that are of uncertain significance. So the Genervon anecdote talks about tongue biting. It talks about fasciculations. Well, fasciculations are not usually bothersome. And fasciculation frequency is not correlated with the speed of other measures of ALS progression, nor are the location of fasciculations telling us anything about where the disease is going to go next. So it's really not clear what a reduction in fasciculation frequency means. This one's probably the most difficult of all. Anecdotes often fail to account for the natural history of the disease, and that's absolutely true in ALS. It took me several years of taking care of patients to understand how different patients can be from each other. It took me several more years to learn that this disease's progression can be very different in an individual person at different times over the course of the disease. And so to prove this, my colleagues and I went into a large internet database called PROACT. And we focused on hundreds of patients who had participated in previous clinical trials and been assigned to the placebo group. And we looked at one outcome measure, the one that's most commonly used in clinics and in clinical trials. It's called the ALS Functional Rating Scale, revised version, also known as ALS FRS-R. And so here in the upper right-hand corner of this slide, you can see one patient's ALS FRS-R score over 600 days under study. this clearly does not look like a straight line for this patient. There are periods of time when they don't progress at all, from day 400 to day 550. And then from day 550 to 600, looks like they're actually regaining some lost function. If we look at everybody in the database, that's what this graph is about. These different curves represent different amounts of ALS FRSR stability or improvement lasting different amounts of time. So for example, this blue line, the Duke blue line, that's the percentage of people in the database that had a stable ALS FRSR lasting different amounts of time. And so, there's a lot of people whose ALS FRSR score was stable for 30 days, but it turns out about 10% of people had an ALS FRSR that was stable for 360 days in that database. If we want to use this kind of information to try to interpret that Jenner von anecdote, we can draw a line here at 6 weeks. Because the Jenner von anecdote said on GM604, that patient was stable for 6 weeks, and that's evidence that the drug works. Well, at least in the PROACT database, 70% of people who were on placebos had a period of time where their ALS functional rating scale was stable. So to me, all that's saying is the natural history of the disease is highly variable. That type of information can't be used to prove that a treatment works. Now, if you're out there and you didn't recognize some of those flaws, don't feel bad. Most non-scientists do not recognize the flaws in anecdotes. And in fact, most non-scientists feel like anecdotes are just as good as the information that we get from randomized, double-blind, placebo-controlled trials. Why is that? Well, no one knows for sure, but this Scientific American article has a theory. It says we've evolved brains that pay attention to anecdotes because false positives, believing there's a connection between two things when there's not, that's usually harmless. But false negatives, believing there isn't a connection when there actually is, that mistake might take you out of the gene pool. Regardless of the reason, it's clear that people are probably overestimating the potential benefits of Aots from anecdotes. And at the same time, I think they're underestimating the potential harms. So there are many examples of individual patients being harmed by Aots. Financial harms. Some of these things cost hundreds, some cost thousands, some cost hundreds of thousands of dollars. Physical harms. There are examples of people who got infected from something they were injected with, people who got blood clots from something they took, people who had tumors and even deaths from different treatments. Now you might ask, how does somebody get a tumor? Well, interestingly, there are stem cell clinics out there that don't have a lot of oversight. And there are now multiple examples, 2 of them on the left-hand side of the slide here, where people actually went to one of these clinics, paid a bunch of money to get injected with what they were told was stem cells, and came home, and not only did they not get better from their illness, but they started to get a new medical problem, headaches, back pain, etc. And when that was worked up, they were found to have a new tumor. And when the tumor was biopsied, it was found to be someone else's cells, not the patient. In other words, they went and paid to be injected with precancerous cells somewhere. That really happens. And then there's one other type of harm, and this one should bother all of us, scientific harm. So we don't enroll very well in our ALS clinical trials. We enroll about 2 patients per site per month over the history of all of our ALS clinical trials. And if you think about it, I mean, busy clinics like mine, we might see 60 to 80 patients in a month. So that's a tiny percentage of all the patients we see. Now, there's a lot of reasons why we don't enroll better. The trials are hard to get into. There's not enough trials. Some of the trials are not very attractive to patients because they require lots of in-person visits, or they require randomization to a placebo for a long period of time, or the results aren't going to be available for years. But certainly one of the reasons that otherwise eligible patients, like Samantha, might turn down enrollment in a trial is because they've decided to pursue an AOT based on anecdotal data. And so, frustrated by all this, in 2009, I got together with a group of clinicians and scientists who also were feeling the same frustrations. And we put together this program called ALS Untangled. The goal of this program was going to be to try and systematically assess the different AOTs that are out there that patients are considering, and ultimately help them make more informed decisions about which ones they would try. So it's kind of like the motto of the X-Files. The truth is out there, but we know it's not always easy to find. And this has evolved over the years. There are now 3 parts to it: inputs, investigations and reviews, and finally outputs. So the whole program is organized on the website, www.alsuntangled.org. When it comes to inputs, these are where we get our ideas. Well, they come from you. They come from patients and families. It's a slow week when I don't get at least 2 new ideas from a patient or family member. And these either come to me in patients that I see face-to-face, or they come via my email directly from a person living with ALS, or they come from a colleague at another clinic who had a patient ask him or her about something that we haven't reviewed. We had to create a place to put these. And so as soon as we get one that we haven't heard about before, we put it on the future reviews section of our website. And if you go to that section of our website, you'll see that we have over 500 AOTs that patients and families have asked us to review. That's more than I will probably ever get to in my entire life. And so we have got to find a way to prioritize these. And we came up with sort of a 2-sided system. The main way that we prioritize things is by votes. So anybody in the world can go on there and look down that list. And if you see things on there that you're interested in us reviewing, just click the vote button and that thing will move closer to the top of the list. And we try to focus on the things that are near the top of the list. But we also had to add a multiplier. And that's because there are some things out there that someone might be interested in, but there might be absolutely no useful disclosable information that we can write about. And so in that case, the votes will be multiplied by 0 until such time as something that we can write about appears, and then the multiplier is a 1. If there's any clinical trials out there on that particular AOT for any condition, then the multiplier is 2. And that's because we get the most information from clinical trials. We also had to come up with a way to review these, and this had never been done, and still to my knowledge isn't being done in any other disease space other than ALS. So we had to invent this. And the first thing I wanted to do was put together a team. Fox Mulder put together a great team, and I'm blessed now to have a huge team, 120 clinicians and scientists from across 11 different countries are now working on this program together. And we had to invent standard operating procedures to try to make this as objective as we possibly could. And so these include, how we get information, specifically how we write these drafts, how we crowdsource them, and ultimately publish them. And when it comes to gathering information and writing, it all revolves around something that we built called the table of evidence. So for each one of these AOTs, we want to pay attention to 5 different categories. Mechanistic plausibility is the first. What is this thing supposed to do? And does that make any sense for ALS? Second is preclinical models. Was this thing ever given to any sort of cell or animal model of ALS? And if so, what happened? The third, case reports. So how many people say they tried this? Can we actually find those people? Can we get their medical records? Prove that they had ALS, prove that something good really happened to them while they were trying this. Trials, and then finally risks. What's the likelihood that something bad or really bad could happen to you if you tried this? And in each of those 5 categories, we assign a letter grade. And there are very specific cutoffs for getting the next higher letter grade. Obviously, the best grade is A, the worst grade is F. But to give you an example of how this works, for case reports, if all we can find is one person somewhere on the internet saying that they bought this product, they tried it, and it made them better, but we can't actually ever communicate with them or get their records, then that gets a D. If we can find that person and get their records and prove they had ALS and that something objective really did improve, gets a C. If there's more than one person like that, it gets a B. And if there's many of these people published in a peer-reviewed article, then it gets an A. So once we've written the article and crowdsourced it across the team and sent it into a journal, it gets one last independent peer review. And once that peer is satisfied, it gets published in a journal, the one called ALS and frontotemporal degeneration. And all the articles that we publish in this journal are published through an agreement called free open access. That means no one ever has to pay to read these. They're also published on the completed reviews section of our website. So if you open up that section, you can see all the different names of the treatments that we've reviewed on the left-hand side of the page. And if you click on the name, it will open up the review. So you can read the whole thing if you want. If you don't feel like reading the whole article, you just want to see what we thought about it in terms of the grades, those are right there on the page. Some of our reviews are getting pretty old now, and so we've started to update them. If you're on that page and you see that the name of an AOT has a star next to it, that means that we've updated it since it was published. We also know that not everybody wants to read their information. I know I'm a big fan of podcasts now. I love to listen to podcasts when I'm driving or traveling. And so we've partnered with a group called the CREATE Consortium, an NIH rare disease clinical research network centered in Miami, to do podcasts. And so a family member, a surviving spouse of a person with ALS, reads these articles and asks me questions from the standpoint of a non-scientist about these reviews. And that's what these podcasts are. And so on that completed review section of our website, if you wanted to listen to a podcast, all you have to do is click the little microphone button, and it will take you to this page where there's a list of all the podcasts that we've done, and you can just click play. We've also learned in the last few years that people in other countries are interested in these kinds of things, these AOTs, and they want to know what ALS Untangled thinks of different topics. And so we've now got a group in Italy that is translating all of our podcasts into Italian. And most recently, we found a person within the International Alliance of Motor Neuron Diseases who's translated our entire website into Spanish. So if you'd rather read about these different products in Spanish, on the main page of our website, you'll see a little American flag up in the right-hand corner. If you click on that, you can change it to a Spanish flag. So this has been a tremendous amount of work. Since 2009, there have been very few days when I haven't spent at least an hour or two investigating an AOT or trying to make this program more user-friendly. Each one of these reviews takes me personally about 40 hours. It would be reasonable to ask, is all that effort really worth it? Is this program a success? Well, I can tell you that people are at least reading our reviews. So the journal that we publish in, they keep track of our most downloaded reviews in the history of the journal. And ALS Untangled has 8 of the top 10 most downloaded reviews in the history of the journal. In fact, we got all the top 4. Some of our individual reviews have over 40,000 downloads. Collectively, the reviews have over 200,000 downloads. Podcasts have over 15,000 downloads. Our peers within the American Academy of Neurology like this program so much that they awarded us the 2019 Brainstorm Award for the best use of technology to improve patient care. But my favorite part of ALS Untangled is all the things that I have learned from it. So first, I'll tell you about some of the lessons I learned about the proponents of AOTs, the people that sell these things. And I'll be honest, when I started this program, I kind of thought of these folks as the Wild West snake oil salesmen. And what I've learned is the motivations of proponents vary. We certainly have come across some snake oil salesmen. The worst of the worst was this guy, Larry Stowe, who once upon a time was running something called the Stowe Morales Protocol down in Mexico. It was a concoction of supplements and stem cells, and he had medical degrees on his wall, and he was telling people that they would recover from ALS if they used his protocol. Well, 60 Minutes got involved, and they went down there and investigated him. It turned out he actually didn't have a medical degree. Those documents were forged. And he later denied making any claims about, the efficacy of his protocol, even though 60 Minutes had them all on hidden camera. And after that 60 Minutes program about him aired, he disappeared for a while. He was finally captured and he's now in prison. But I would say most of the people that I've met who sell these products are not like that at all. Most of them are what I would call true believers. And here's one of those folks. This is Dr. Joseph Hickey. He runs a wellness clinic in Hilton Head, South Carolina. And I reached out to him because a patient had asked about this. And he invited me to come down and spend a couple days with him and his wife and see them in action. Now, how do I know that Dr. Hickey was a true believer? Well, because when the clinic closed, he and his wife would chelate each other. Their whole hypothesis was that metals cause diseases like ALS. So do I agree with that? No, but I do believe that Dr. Hickey believes it because I saw him and his wife treating each other for heavy metals to try to prevent diseases like ALS. The other thing I learned about Aots is they do some things really well that mainstream doctors need to pay attention to. So, I talked to a lot of his patients in between their treatments, and I asked them, tell me how you got here. And their stories were remarkably similar. They would tell me, well, I went to this famous clinic or that famous clinic, and I felt like it was all gloom and doom. It was all just measurements that were worse and difficult conversations. And I just felt like I didn't get any hope. And when I would suggest something that I saw on the internet, it would just be shot down without really any kind of discussion. And I would say that Dr. Hickey was the polar opposite of that type of doctor. He was incredibly hopeful, incredibly optimistic with every patient. He was very open-minded and he was very responsive. Even after a long clinic day, he and his wife would stay there until every phone call and email had been answered before they went home to have dinner. And I tell young doctors now, look, if you're gonna be an ALS neurologist, you've got to embrace these 3 personality traits. You've got to find a way to be optimistic for your patients. They need hope. You've got to find a way to be respectful of their ideas. That doesn't mean you don't discuss them and help them to investigate them and have an opinion on them, but you've got to be respectful. You can't just shoot down people's ideas without even looking at them. And finally, you got to be responsive. When people have questions or problems, they expect you to get back to them in a timely manner. The next group of lessons I learned were about the specific Aots. And again, I'll be honest, in 2009, I thought most of these things were going to be as bogus as this picture of Bigfoot that's emerged. And we certainly have found some Aots that I would say are less promising. And to try to help people identify less promising Aots, we collected some of the things that were associated with Aots we reviewed that we didn't give very good grades to. We call these the red flags. So watch out if somebody is trying to sell you something that's unproven, that costs a lot of money. It's really unethical to do that. When we're testing something in a clinical trial, we don't charge you to take the product. We have a budget. In fact, many times we'll pay you for your time and for your travel. Watch out for things that are advertised as effective for multiple incurable conditions that seem to have different causes. Some of the most bogus Aots we looked at, there were 30 or 40 things that they were advertised to be able to cure. Do you really think if someone had the cure for those 30 or 40 terrible things that they would have to have their website set up in some dark corner of the internet? I mean, they would be the most famous, wealthy person in the history of medicine if they really could cure all those things. Watch out for things that don't have safety and scientific oversight. Watch out for things that don't have a very detailed, informed consent process. Watch out for things that don't have a very scientific mechanism by which they might help. If you're going to study something, you're going to give people something that's unproven, you ought to at least measure something afterward. And so watch out for clinics and doctors who are experimenting on you without actually measuring anything afterward to see what happened. Watch out for people who may be measuring things but don't have any plan in place to ever report the results of those measurements. Scientists, we collect information and then we report it. We report it at meetings. We publish our findings in journals. That's called peer review. And it's not easy. It's not easy to work hard on something for 2 or 3 years and then have one of your colleagues pick it apart, but that's how you get better. And that's how science moves forward, by people trying to, professionally criticize each other's work. It's part of good science. Watch out for things where the only evidence of benefit is anecdotes. Watch out for things that are being offered to you by someone who doesn't really have any relevant medical or scientific background. And finally, watch out for things that are being offered by people who portray themselves as victims and advise you to get rid of your mainstream doctors. The mainstream doctor may not be perfect, but they know a lot. They've worked hard to try to help you with aspects of this disease. And even if you decide to pursue something that your doctor doesn't agree with, that doesn't mean that everything that your doctor is telling you is worthless. I mean, sometimes people have a hybrid model where they work with somebody who's selling these kinds of things, but they also work with a mainstream doctor, so they get the best of both worlds. But some of these Aots, it turns out, are very interesting and I believe warrant further study. So there's several of them that have plausible mechanisms by which they could help a person with ALS: acetyl L-carnitine, cannabis, curcumin, melatonin, methylcobalamin, protandim, resveratrol, tamoxifen, vinpocetine, butyrates. Several of these things do have positive preclinical data from ALS models. Again, acetyl L-carnitine, cannabis, curcumin, melatonin, resveratrol, tamoxifen, butyrates. And believe it or not, some of these Aots even have clinical trials in people with ALS that were positive. Now, none of these were perfect trials, to be sure, but acetyl L-carnitine, basis, curcumin, hyperimmune goat serum, L-serine, methylcobalamin, tamoxifen, and TUDCA all have clinical trials that suggested that people who took these products did better than people who didn't. And then maybe the most dramatic thing that I've learned in all these 13 years leading ALS Untangled is Nelda Buss, the woman in that video that I showed you in the beginning. She's real. So I never in my wildest dreams imagined that this could happen to a person with ALS. But I found Nelda Buss, I talked to her on the phone, and she was so kind. She was kind enough to send me a box of her medical records. And after reading those records, I'm absolutely convinced that her ALS diagnosis was correct. She saw some of my most trusted colleagues at the University of Virginia. She had the right story, the right neurological exams, the right EMGs, tons of testing to rule out ALS mimics. She really appeared, by today's definition, to have ALS. She really did progress, as documented in those notes, to where she was paralyzed and quite near the end of the disease. And then, unexpectedly, she recovered. Over the next 2 years, she appears to have completely recovered all of her lost strength. And all of this happened, by the way, 30 years ago. She's still alive, and she still appears to be neurologically normal today. And so when I discovered all this, I had to ask myself, what do I do with this amazing case? Never heard of anything like this. Well, to me, it was immediately clear that it was worth studying. There is a precedent in other areas of medicine for people who do unexpectedly well with some disease. So, for example, in the HIV world, we've known for more than 20 years about a group of people who get infected with the HIV virus but never get sick. They're called elite controllers. It's about 1% of everybody who gets infected. And it turns out, for a long time, people just shrugged their shoulders. And then somebody said, we need to study these people and see what's different about them. And many of them have a genetic abnormality. It's in a gene which is called CCR5. Their abnormality is called Delta 32. And when they recognized this, they said, well, what is this gene doing that might be relevant in the biology of HIV? It turns out it codes for a protein in the surface of cells that HIV needs to get in. And in these people, that protein looked different. And HIV couldn't use it to get in as well. And once that was discovered, Pfizer jumped on that discovery and built a drug that blocks that receptor. And it works for everybody with HIV. So to me, it's definitely worth studying cases like Nelda's, cases of ALS reversal, because maybe I can find out something different about them that would allow me to reverse ALS in everyone. Now, if you're going to study something, you've got to first define it. And so there's 3 parts to how I define an ALS reversal? First, I've got to have enough medical records to be able to independently confirm the ALS diagnosis. There's got to be the right history, the right exam, the right EMG, and the right testing for mimickers. Second, there's got to be progression of that patient's weakness to the point where they had some disability. And then third, there's got to be a dramatic and persistent recovery of lost motor function and at least some disability. And so as of today, I have 56 examples that meet that definition that I call dramatic ALS reversals. And, just to give you some examples of what happened to these people, some were ventilator dependent. Now they breathe on their own. Some couldn't swallow. They needed a feeding tube. Now they swallow normally. Some couldn't speak. Now they talk normally. Some were wheelchair bound. Now they walk. Some can even run. Some were completely dependent on others for their activities of daily living, and now they take care of themselves. And so the next thing you got to do if you want to study something after you define it is come up with some hypotheses. And I would say there's probably 3 hypotheses for these cases. The first is maybe these people never had ALS in the first place. Maybe they had some kind of mimicker. The second is maybe there's something different about these people that allowed them to beat the disease, kind of like the HIV elite controllers. And the third, maybe these people found some things that actually worked against ALS. And so this is why I don't think that these cases are mimics. I've looked at the medical records of these folks. They have classic stories. They have classic exams. They have classic EMG testing and classic testing for mimics. Several of them have a family history of ALS. About the same percentage of these folks have a family history as we see with more typically progressive ALS. So if these folks had something completely different, why would roughly 10% of them have a family history of ALS? It just doesn't seem to make sense. And then finally, if you look at how these people recovered their strength, it wasn't magic. They appear to have recovered from something called re-innervation. And so let me explain to you what that means. This is an example of what happens to a person with polio. And let's say that this thing that looks like a railroad track, let's say that's your biceps muscle. And before polio, you had 4 motor neurons that were innervating your biceps muscle. And then you got polio, and it killed one of the motor neurons completely, made another one real sick, but you survived. And so in this stage, your biceps muscle was weak. It didn't have the same number of connections. But over the years, those 3 now, instead of 4 motor neurons, they actually were able to send out branches, and they were able to reconnect completely with the muscle. And this person regained their strength. But what's interesting is if I stick a needle in this muscle, if I do an EMG on this person who recovered from polio, that muscle's not normal. It shows me signals that are associated with re-innervation via collateral sprouting. And there's a handful of these ALS reversals that actually had EMGs after they recovered their strength. And this is exactly like a person with post-polio syndrome who recovered. So even though the muscles were full strength, they were not normal. There was evidence of re-innervation from collateral sprouting. So the first program that I have to study a group of hypotheses is called STAR, Study of ALS Reversals. I'm trying to find as many of these cases as I can. I'm trying to put them all into the same database for the first time in history, try to compare their demographics, their disease characteristics, their comorbidities, the things they took to people who are in databases of more typically progressive ALS patients. I'm trying to get biological samples on as many as we can, and I'm trying to ask them as many questions as I can about their lives and the things they were exposed to. And so far what I know is that these ALS reversals, they tend to be a little younger than your typical progressive ALS patient. They're a little more likely to be male, a little more likely to have limb onset disease. They seem to progress a little faster before they start to recover. And they appear to be more likely to have taken certain Aots. And this is a table of all the Aots that were significantly more likely to be associated with an ALS reversal than they were with people in databases of typically progressive ALS. And you'll see at the very top of the list, with an amazing odds ratio of over 300, is a product called Curcumin, which we will talk more about. I have several collaborations underway to study these people in different ways. We're looking at the pictures that have been taken of their body with Duke Neuroradiology. We're looking at their genetics with the CREATE Consortium, and we're almost ready to release that information. We're looking at the RNA and the proteomics with a group called ALS TDI. We're looking at the microbiome, the family of organisms that lives in the gut with the Duke Microbiome Center. We're looking at the environmental exposures with the National ALS Registry, and that paper is hopefully less than a month from being published. And finally, we're going to be looking at autopsies. If these people ever die, hopefully not for many years from natural causes, we would like to compare their brains and spinal cords to people who die from more typically progressive ALS and see if there's anything different there. And then the last hypothesis, is it possible that these reversals could be due to the treatments that these patients took? This is the one that sometimes raises eyebrows amongst my colleagues, but it shouldn't. There are numerous examples in the scientific literature of patients discovering drug effects that researchers had not previously thought of. This is called serendipitous drug discovery. And the most famous example is Viagra. So Pfizer owned Viagra. They were studying it for people with heart disease, and it didn't work, and they were about to scrap it. When males in the study came forward and said, we'd like to keep getting some more of this if we could, because it seemed to help with our erectile dysfunction. And the scientists who had Viagra had no idea that it would do that, but they quickly switched gears and developed it, and it became the first-in-class drug for that indication. It was a $1 billion blockbuster discovered by patients, not by researchers. Nudexa is another example. We had studied that drug to see if it could slow the progression of ALS, and it didn't. And again, we were about ready to get rid of it when patients in the study came forward and said, hey, this is really helping the problem that we had before, controlling our laughing and crying, what's called pseudobulbar affect. So Nudexa was a serendipitous drug discovery. So the name of this research program that I started is called Replication of ALS Reversals, ROAR. This is a program of small pilot trials of these Aots that are associated with dramatic ALS reversals. Now, because most of these products are regarded by the FDA as safe, as you know, many of these products are just sold on the internet without a prescription. And because I'm looking for such a huge signal, I'm not looking for a 20 or 30% slowing. I'm looking for recovery from ALS in these trials. These can look very different than your typical pharma-sponsored trial. I can have very wide inclusion criteria. Why are pharma trials having such narrow inclusion criteria? Because they're looking for a small signal in a noisy disease. I'm looking for a huge signal, so just about anybody can participate. These trials don't need placebos. If I have people that start the trial and they're wheelchair bound and at the end they walk, no one is going to say that was a placebo effect. I don't need in-person visits for these trials. The things that I'm measuring are so obvious that I can teach people how to measure these things, and I can even teach them to enter the data themselves on a website called Patients Like Me. And the advantage of doing that is not only sparing them the burdens of travel, but now the data is on a publicly accessible database. Anybody in the world can look in at one of these ROAR trials in real time and see if anything interesting is happening. And then finally, because I know these trials will fill up fast, they have filled up fast, I publish these protocols once they're IRB approved on the website, alsreversals.org. And the reason I do that is because, as I said in the beginning, I know that there's people all over the world with this disease that want to try things. And so why not put a roadmap out there for them? If they want to try something that's in this program, they can figure out exactly, what brand and what dose and for how long and what kind of things they can measure with their own doctor. And so we've already completed one of these ROAR trials. That was the Lunasen trial. So Lunasen was a peptide. It was said to be a histone deacetylase inhibitor. It came from soybeans, and it was associated with a really dramatic reversal of bulbar ALS from Providence, Rhode Island. And so we put it into a clinical trial. 50 patients took it for a year. And there was some good news and some bad news. The bad news is the product didn't seem to be as safe as I thought it was going to be. Many patients had side effects, including cases of constipation so severe we had to hospitalize patients. There was no evidence in the biomarkers that we used that this could alter histone acetylation in humans. There was no evidence that it slowed, stopped, or reversed anyone's ALS in the trial. But the good news is that this design worked really well. This was the fastest enrolling trial in ALS history. I told you most trials enroll 1 to 2 patients per site per month. We enrolled more than 9. The people who are in this trial are much more similar to the people that I see in my clinic than the ones that are in most clinical trials. For example, we had some people in this trial that had had ALS for 10 years. We had some people in this trial that were using ventilators and feeding tubes. And in spite of taking people that were further along in their ALS journey, they stuck with us just as well as people in more traditional trials. 84% of everybody that was still alive at the end of our trial was still entering data. And we found out that we empowered hundreds of people around the world who couldn't get into the trial because it filled up so fast to follow the protocol by publishing it. And in that way, we allowed them to self-experiment with something that we at least thought at that time was very reasonable. And so we are geared, we have well underway our next ROAR trial. And as you might have guessed, it's of that AOT that was at the top of the list for ALS reversals, curcumin. Curcumin is a molecule that looks like the ones here in these diagrams. And in real life, it's a bright orange powder. It's actually found in spices like turmeric and curry powder. It's got a lot of ways that it could help a person with ALS. It's anti-inflammatory, it's antioxidant. It interferes with the clumping of proteins that we think is important in ALS. It also may be able to alter the gut microbiome, which is a really hot topic in ALS right now that I'm excited about. There have been positive preclinical and even clinical trials of curcumin in different countries. There are now 9 ALS reversals associated with curcumin products, and it appears to be safe, at least when taken orally. And so I've got a ROAR trial of a specific form of curcumin underway called Therakenum. This one also filled up very fast. We've got 50 people with ALS in this trial. And the last one is supposed to finish, I believe, at the end of next month. And so we hope to have results to share with you in July or August. But all the patients in the trial are getting the real product, Therakenum, at a dose of 1 capsule twice a day. And we're looking at their progression versus historical controls. We're also looking to see if we can alter the gut microbiome in this study. And again, there's no in-person visits. This is all virtual. It's all done on patients like me. And the results are available in real time. And the protocol is published on the ALS Reversals website. And so to just kind of summarize what I told you today, when it comes to these things called AOTs, the truth is out there, like they used to say on the X-Files, but it's not always easy to find. Anecdotal data is especially problematic in ALS, and I think it's commonly misinterpreted by non-scientists. We've got this group called ALS Untangled. It brings together people with ALS, their families, clinicians, and scientists from all over the world to try to systematically review and report on AOTs. And we can help you to validate and centralize both positive and negative anecdotes and to temper conclusions based on what we know about the natural history of this disease. But there are some anecdotes that I think are worth paying attention to. That are these 56 ALS reversals that I've validated. These are rare, but I think they could be really important. They could provide us with clues to novel mimics, endogenous mechanisms that fight the disease, or even new treatments. And we now have a definition of these. We've got testable hypotheses. We've got collaborations with researchers all over the world underway for studying these. So I'm very, very excited about this. And so at the end here, let me just make sure to thank all the people with ALS that are out there. You probably know this by now, but you're the reason I get out of bed every day, and I'm so excited to work on all these programs. You inspire me. Thanks to the ALS Association, the MNDA, the LVH ALS Foundation, all of our other funding sources. We couldn't do all this work without funding. Thanks to our collaborators at Patients Like Me, Neurobank, Origent, and all the reviewers around the world who volunteer their time to help me with ALS Untangled. And finally, thanks to my Duke ALS care team for helping me build a great foundation of patient care upon which all my other ideas, all my research is built. So I'll be happy to stop here and take questions from you.
Thank you so much, Dr. Bedlak. That was terrific, as always. So we have a few questions. People can continue to enter questions. We are going to be running up against the clock, but we can go over by a few minutes. So the first question we have is, what do you think about chelation therapy? You did mention it in your presentation.
Yeah, so that's what was happening at the Hickey Wellness Center. Their theory was that metals cause ALS, and that a good treatment would be to have your fillings removed and to undergo chelation, which is basically to be infused with a chemical that pulls metal out of your body. So I have not seen convincing evidence that there's any metal that causes ALS, nor have I seen any evidence that having metals pulled out of your body can make a person with ALS better. There definitely is evidence that doing that can make someone very sick. So at the present time, when my patients ask me about it, I say, I actually don't think that's a very promising AOT chelation.
Thank you. The next question is, isn't this neuroplasticity? This is discussed in many neurological diseases, not just ALS reversals and stoppage of progression.
Yeah, so it is a form of plasticity. Usually when we think about plasticity, we think about it happening like in the central nervous system. Like for example, after someone has a brain injury when they're real young, you know, the central nervous system, especially in young people, is very plastic. It can remodel itself. But yeah, the peripheral nervous system can do that all through our lives. The reason I put that in there was just to say, you know, when people look at me and they say, well, these people, you know, that this is just some sort of, you know, miracle thing that they recovered from ALS. I don't think it is. I mean, I think there's a precedent by which people can recover from another motor neuron disease, polio. And so, I mean, if they can recover from polio by wiping out the polio virus and gaining their strength back through this plasticity, or what I called collateral sprouting, why couldn't that happen to a person with ALS? I mean, why is it not possible that somebody could have had something that they could have beaten, some cause for their ALS that their body beat, or that some product they took beat, and then they recovered through the same process, plasticity or collateral sprouting?
Nice. Okay, next question is, what do you think is the cause of very slow progression in some cases of ALS?
Yeah, so right now, at the top of my list of possibilities, I think it may be something about the microbiome. So the microbiome is this family of organisms that live inside our guts. There's trillions of them. And you know, we've known for decades that alterations in that family can cause diseases of the gut, like inflammatory bowel disease or C. diff colitis. But in the past decade, it's becoming more and more clear that alterations in the microbiome can influence how people do with other diseases, including brain diseases. And the best evidence for this in ALS is we have different animal models where we insert abnormal genes into, you know, mice and things like that. And those animals develop, you know, a progressive motor neuron disease that shortens their lives. So simply by altering the composition of the microbiome in those models, people have shown the ability to dramatically slow down or speed up the progression of the disease. That's extraordinary. Now, how does that work? Well, there's something called the gut-brain axis. It's quite possible that the microbiome is secreting something. In some cases, something that might be neuroprotective, like butyrate is high on my list of possibilities, or in some cases, something that might be harmful. So it's possible the microbiome could secrete something that ramps up neuroinflammation. It's possible the microbiome could secrete toxins like beta-methylaminoalanine. And so we now at Duke have underway a study looking at the microbiome of people with really slow progression, really fast progression, and ALS reversal. And we're hoping that there's a difference. It would be great if there was just one thing that was really high in people with reversals and, you know, a little less in people with slow progression, and not there at all in people with fast progression, because then we would know exactly what to do. We would build a probiotic to give people that one thing in the highest possible amount in their microbiome. But it may be more complicated than just one thing, but that's what I'm most excited about now as a cause for the differences in progression, the microbiome.
So you don't think it's two different diseases with similar symptoms?
I do not think this is a different disease. I think the disease can be modified by a person's genetics and their environment and things like that. But I do believe, you know, this idiopathic degeneration of motor neurons, it may have different causes, it may have different modifiers, but I think calling it the same thing makes sense. That doesn't mean that we're going to treat it the same way someday. We may be treating, subtypes of ALS in different ways.
Okay, next, how do you determine who would be in your Therakenum study?
Yeah, so the first 50 people who qualified are the ones who got in. We did have, unfortunately, a waiting list of about 250 people. When we said that study was nearing opening, 250 names on a list, and we just took the first 50 on the list that qualified. And it's kind of like what we do for every trial is we keep a list of, once we start getting close to opening, we kind of announce it, and then as people contact us, we start keeping a list of who wants to do it. I don't know of a fairer way, but at least for these trials, if you didn't get in, the protocol is published, and you can take that protocol to your own doctor and see if your doctor thinks that would be a good thing for you to try while we wait for the results.
Oh, thank you. Is there a webinar to help me learn to navigate ALS Untangled?
Well, that's a great suggestion. I can certainly make that suggestion to the Northeast ALS Consortium, Niels. We haven't done an ALS Untangled webinar in a long time, but based on that question, maybe we need to actually have one where we start right from the beginning, from the home page, and just in real time, walk through all the different things you can do with that website and answer questions about it. So thank you for making that suggestion. I'll reach out to Niels today.
Thank you. Can you speak to your thoughts on AMX0035, a butyrate and supplement, and thoughts on use of combination drugs for treatment?
Yeah. Well, as I mentioned, I think that there may be, although I think ALS is one disease, I think there may be lots of different ways in subtypes of ALS that motor neurons die. And I doubt that every person sitting in front of me in clinic next Tuesday is going to have the exact same mechanism killing their motor neurons. I bet you some people have more neuroinflammation, and some people have more mitochondrial dysfunction, and some people may have more of a toxic cause. And until the day comes when we get biomarkers that can tell us who has what, the next best thing is to develop cocktails of treatments. So we got Riluzole, that works on something called excessive glutamate. We got Radicava, that works on something called oxidative stress. AMX0035, the two components of that, they work on other things. PUDCA works on mitochondria, and sodium phenylbutyrate, the other ingredient that's in there, works on something called histone acetylation, which I mentioned earlier. We know that there's an abnormal pattern of genes that are turned on in people with ALS. It's actually a destructive pattern instead of a protective pattern. And part of the reason for that is abnormal histone acetylation. If we could somehow normalize that, we might get those protective genes turned on, and people may be able to help heal themselves. And so I really like the mechanism of action. I think the trial that was done was the best kind of trial you can do. Is randomized, double-blind, placebo-controlled. We had fantastic oversight. We had great sites participating in the study. I think the results are very believable, and the results are important to people with ALS. I mean, the drug showed a 30% slowing in functional decline, and it showed a significant prolongation in survival, prolongation until the time that someone needs to consider a ventilator, prolongation to the time when someone might be hospitalized for the first time. Those are all meaningful outcomes. It's the first drug that's actually shown the ability to affect more than one outcome in ALS, and all those things happened on top of Riluzole. So I think that, if that gets approved, which I hope it will, then we'll have a cocktail of things, and hopefully the cocktail will grow as new drugs come out with even different mechanisms.
Okay, so I've been given the cutoff time sign. So I'm going to give you one more question. Try to make it in less than a minute answer. Of the 56 reversals, can you pinpoint any AOT they all of 90% of them took?
I wish I could, but so far I haven't found a single thing that they all appear to have in common. As I said, curcumin products are the ones that were used by most, and that's 9 of 56. So there's no supplement or product or medication that was used by more than 9 of them.
Okay, terrific. I think we're going to have to ask you some of these questions later and ask you to please get back to us on them. But thank you so much for your time. We do have another session starting at 2:15. There are 3 different sessions up against each other, 3 different breakout sessions. So I hope everybody joins those sessions. And Dr. Bedlack, thank you so much for your time and your spirit. We appreciate you here and everywhere in the ALS community.
Thanks, Teresa.
Have a
Good
great
night, everybody.
day.
You too.
Enjoy your swim.